we see a neuron as a computer in its own right, but maybe even a supercomputer, because individual cells have their own intelligence, and there's a lot of work showing that a single cell is able to perform quite complicated cognitive tasks.
The Rational View is a weekly series hosted by me, Dr. Alan Scott, providing a rational, evidence-based perspective on important societal issues. Hello and welcome to another episode of The Rational View. I'm your host, Dr. Al Scott. This episode continues my series on consciousness. Are we just biological robots? Following on from my exciting interview last week with quantum biophysicist Dr. Luca Turin, I dig further into competing theories of quantum mind, or consciousness mediated by quantum mechanics, the physics underlying the very small. The famous Sir Roger Penrose and his partner Stuart Hameroff have proposed the Orchestrated OR theory. This theory posits a modification of what is known to be the incompleteness of quantum mechanics that underlies half of physics. They argue that consciousness arises from moments of wavefunction collapse that are objectively linked to certain aspects of quantum superposition. So they're postulating an update to the theory of quantum mechanics, which is based specifically on quantum mechanics being linked to consciousness. So it's a physical theory of consciousness related to quantum mechanics. They further argue that biology has evolved a mechanism to orchestrate these wave function collapses in tiny subcellular structures called microtubules, and they present some criteria for this to happen, and experiments are ongoing to determine whether that's real or not. My guest today has been doing experiments in the lab to determine if physical processes in subcellular organelles called microtubules respond to anesthetics, and then may potentially be linked to this altered quantum mechanical theory of the mind. If you like what you're hearing, please press like on your podcast app and share it with your friends. Love to hear from you. Come check out my website at www.TheRationalView.ca. Professor Jack Tuszynski obtained his PhD in condensed matter physics in 1983 from the University of Calgary. From 1983 to 1988, he was a faculty member at the Department of Physics of the Memorial University of Newfoundland in St. John's. He moved to the University of Alberta in 1988 as an assistant professor. Between 1990 to 1993, he was an associate and then full professor at the Department of Physics. Between 2005 and 2020, he has held the prestigious Allard Chair in Experimental Oncology at the Cross Cancer Institute, where he leads an interdisciplinary computational drug discovery group. He is also a fellow of the National Institute for Nanotechnology of Canada. He has held visiting professorship and research positions in China, Germany, France, Israel, Denmark, Belgium, and Switzerland. He's widely published in the scientific literature, providing frequent invited talks on his groundbreaking work. Professor Tuszynski, welcome to The Rational View.
Pleasure. Thank you for having me.
Thank you so much for coming on. Could you tell me a little bit about your background? You're from Poland originally, I take it? How did you come to Canada?
I was born and raised in Poland, and I did my undergraduate studies there. And then it so happened that there was a lot of political, social turmoil in Poland around the 1980s. And I decided to leave that country because there was, at that time, actually not much future for me. And so I went to Canada to do my graduate studies. And I stayed.
Was this while it was the Soviet Union that you left?
Well, I never actually visited the Soviet Union, but it was the threat of the Soviet Union encroaching on Poland that it was at that time part of the Warsaw Pact.
Mm-hmm. Can you tell me a little bit about your current research work? You've obviously very broad disciplines here. You've got physics, you've got oncology, you've got nanotechnology. Tell me what your career path was and how you got into these diverse topics?
Yeah, I think it's first of all, I would like to say that a degree in physics is equipping students with a lot of tools in the toolbox, scientific toolbox, allowing you to study different things and make progress in different areas. And so that happened with me. I started with theoretical physics and then gradually moved towards biology, which was always of interest to me, but until more recently, maybe the last 2 decades or so, biology wasn't a field where you could do a lot of quantitative work. And as a physicist, I like to understand mechanisms. I like to develop mathematical models and make predictions about the behavior of biological systems on this basis. So that led me naturally initially to some areas of the life sciences where you can apply physical tools, which actually the first field in this area that I studied was pharmacokinetics. So this is a field of study that informs you about the fate of the drug in the human body, how it distributes itself, where it goes, how it interacts with different cells, how it's metabolized, excreted, and so on. And that was very interesting because I was able to use a lot of fairly sophisticated mathematical tools, including nonlinear dynamics, fractal kinetics, and things like that.
Fractal kinetics is a word I've never heard before.
There you go.
That does sound.
Well,
that's very interesting.
Yeah, quite simple. It's it's about molecules which diffuse or propagate, spread out on surfaces and in structures which are fractal, which means self-similar. And it so happens that the human vasculature is fractal. Just like branches of the tree, leaves, and so on, they have this self-similar nature. So does human vasculature. And therefore, if you want to study the fate of a drug molecule in the human body, that helps you understand how this molecule or group of molecules propagates throughout the various organs over time. So that was the first area, but then other areas opened up gradually to more sort of mathematical, physical considerations, and that included proteins. Proteins are very important for human health and also play a major role in development of numerous diseases. And so I studied some of the most important proteins, such as tubulin and actin and motor proteins. The first two play a role in cell division, very important roles in cell division, cell motility, movement of cells. And motor proteins are basically the muscles of the cells. They also transport material across the cell from point A to point B. So these are, as you can imagine, very physically oriented processes. And so I studied that. And one of these proteins, tubulin and microtubules, are at the core of the question of how cells divide. And that, of course, is one step removed from cancer. And when we want to stop the proliferation of cancer, the first question is how do we stop them from undergoing cell division? And therefore, microtubules and tubulin become natural targets for drug discovery. And that led me to this position in the Department of Oncology, which I held for 15 years. And my group was developing antimitotic chemotherapeutic agents, one of which is now in clinical trials. So I felt I accomplished my mission because we actually achieved something which is tangible. I don't know if it'll be eventually used, in a widespread context, or maybe it's just a trial that doesn't go to the next step. But some humans are being tested. Patients with bladder cancer are currently being tested in the clinical trials.
And how long did it take to get to that stage of clinical trials? How much research went into that?
That's a very good question, actually, because when I moved from physics to oncology, I started by looking at other case studies, other drugs which were developed by big pharma and medium-sized pharma. And until then, which was, I started in 2005, most of it was really by standard sort of trial and error methods, empirical studies. And it took typically between 12 and 15 years. And about the cost of development is about on the order of $1 billion for each entity. And so I was very arrogant. I thought, I might be smarter than that, and we can do it faster and cheaper. And it turned out that we were able to do it cheaper, but not faster. It took almost exactly 15 years to develop this new drug. Actually, it's one drug which is called CCI-001. CCI stands for the Cross Cancer Institute, which is where the Department of Oncology is housed at the University of Alberta in Edmonton. So
wow.
So much.
Well, congratulations! I hope that that pans out and has a tangible effect on cancers worldwide. That's great. So your work has now progressed to you're involved in what I would say that theories of mind and consciousness. The work you're doing now or recently that you've been talking about touches on quantum theories of mind, and specifically the orchestrated objective reduction theory of Penrose and Hameroff. Can you describe a little bit about how you got into that line of work?
Yes, indeed. Well, that was more or less in parallel with my other research, and never really my main focus. It was until recently kind of on the periphery, a hobby of sorts, and partly because it's very hard to get funding for research in this area. But I got really interested, and it started with a very momentous conference, momentous event. In 1991, there was a conference held in Tucson, Arizona, jointly with Stuart Hameroff, and in fact, Al Scott. That's the Al Scott that I mentioned to you. So
Alwyn Scott, you said?
Alwyn Scott, yes. And
amazing.
And Steen Rasmussen. And
we're all over the place.
Yeah, this conference was called "What Is Life," and that was actually for me a transition from physics to biophysics. But at the end of this conference, Stuart Hameroff asked the question, and he said that the next conference should be about something even more mysterious than life. And he said the next conference should be about what is consciousness. And this started from 1992, I believe, maybe 3, I don't remember exactly the date, a series of annual conferences on the science of consciousness. And that's the one that you referred to, I guess, which occurred a week ago. Yeah, exactly a week ago, I think, and last week. And so that's one in a series of more than 20 conferences on the science of consciousness. And I participated in many of these conferences and got to know Stuart Hameroff very well as Roger Penrose, and get interested in this field, and especially because the main object of studies in this case is also the same as I studied for cancer research, namely microtubules. So microtubules play a major role in all eukaryotic cells which have a nucleus, and they do different things in different cells, including cell division, which I mentioned, but also in neurons, which don't divide. They form very regular architectures, parallel bundles with interconnections that remind me and others of computer architectures. And Stuart Hameroff wrote a very prescient book called Ultimate Computing, which was published in 1987. And in this book, he outlined his vision, which actually predated the work with Roger Penrose about consciousness being the substrate of consciousness, the bottom level of conscious experience being based on microtubules in neurons. And so I got really intrigued by this, having spent quite a bit of time studying microtubules. And anyway, that is now 30 years later. We're still doing it.
Wow, that's amazing. So microtubules, these are proteins in the cell that look a little bit like wires, effectively. They're structural elements, and the theory is that these things could be doing processing. So I would say the accepted consensus view is that the basic processing element of the brain is the neuron. And neurons signaling between each other have been simulated in computer science to make neural networks, which are the basis of artificial intelligence models right now, and they seem to work in sort of processing ways. The theory of Hameroff that you're describing and Penrose posits that even inside each cell there is significantly more processing going on than one might expect in these microtubule proteins based on tubulin molecules. Could you maybe summarize the orchestrated objective reduction theory at some sort of a high level for the layman kind of?
Yes, of course. So let me backtrack a little bit and go back to what you said about neurons. And we have almost 100 billion neurons in the human brain on average. So this is massive. And as you said correctly, in current neuroscience, the neuron is considered to be the fundamental element of all the cognitive processes, right? But I think it's a major insult to the neuron because the neurons are very complicated and they have a lot of complex structure, which must be for a reason. Just looking at this, it makes you wonder about it, and I'll come to it in a second. But on top of that, there is, on average, about 10,000 interacting neurons with one neuron. So it's not just a lot of neurons, but a lot of communicating neurons with other neurons. And so that gives you a humongous power. And these are not passive elements. Neurons behave differently depending on their location in the brain, depending on their usage, activity. This is called reinforced learning or long-term potentiation. So if particular neurons are involved in a lot of activities, let's say you learn a foreign language, then you reinforce these interactions and sometimes even recruit new neurons to interact with those. So this is called synaptic plasticity. So you couldn't really think about it as a bunch of wires with switches, binary switches on and off. That's more or less what projects such as the Human Brain Project in Switzerland are envisaging. We go deeper, we meaning Stuart Hameroff and Penrose and other people in this group. We see a neuron as a computer in its own right, but maybe even a supercomputer, because individual cells have their own intelligence, and there's a lot of work showing that a single cell is able to perform quite complicated cognitive tasks, information processing, reaction, and I can point to the work of Günther Albrecht Bühler at the Northwestern University in Evanston, in Illinois. He actually has a major body of work called Cell Intelligence, indicating that individual cells have mechanisms of perceiving environment, reacting to the environment that they are in, and interacting with other cells. And under controlled conditions, he demonstrated this. So that's number one. Number two, I also would like to point out the importance of the work of Mike Levin at Tufts University in Boston, who has been working on reprogramming cells via electrical and electromagnetic means, turning stem cells into different types of cells by electrical signaling, and also looking at the ability of individual single cells to have memory and very primitive organisms without the nervous system being able to store memories and
Wow.
react. I want to say that the brain is obviously one of the most complicated structures in the universe, but you need to look deeper into not just the structure of the brain, but the structure of neurons, which are, by the way, not the only cells in the brain, but the ones which are excitable, that react to action potentials and electrical propagation of signals. And that also brings physics to it, because before we go into quantum physics, there is also a lot of electromagnetic physics which takes place in neurons and in the brain. So now back to your question. So if we assume that the neuron is a non-trivial, non-passive, but dynamic computing element, then the question is, what is computing? Where is this computing taking place? And we know for a fact that signals propagate along neurons' axons, right? So axons are the long protrusions that have membranes, of course, and membranes have ion channels in them, so they actually pass ions in and out and propagate voltage changes. In other words, electrical signals at fairly impressive speeds on the order of about a meter per second. So that's maybe not... it's faster than chemical signaling, but it's not instantaneous. And I think if you were to use this...
It's not a wire.
Yes, exactly. Much slower than electrical signals in electrical devices and so on. So in order to explain some of the more impressive feats of human achievement in terms of, for example, tennis players serving tennis balls flying at 190 miles an hour, and the opponent is able to perfectly return that serve. I don't think you can explain it with this kind of signal propagation and of course delays as well. Right? So there is something else going on. I'm just building a case for something more impressive than just a bunch of ions moving up and down the axon, and also saying that the neuron itself has this complicated architecture. And why? Part of it is, of course, for material transport. You have to provide nutrients and build neurotransmitters inside and pass them to the synaptic batons and stuff like that. But what Stuart Hameroff and Roger Penrose later on noted is that microtubules would be ideal substrates for some deeper computation, a deeper level computation, and perhaps even non-algorithmic computation, which would involve quantum states. And that's where the story of OR begins. So Stuart Hameroff contacted Roger Penrose because Penrose wrote this famous book, The Shadows of the Mind. So in this book, Penrose proposed a quantum nature of human consciousness, and gave a lot of examples why it should be quantum and non-algorithmic. Many examples where he gave many examples where you couldn't explain the outcome of human cognition with a simple algorithm that could be programmed by present-day computers. And that led him to believe that this must be a form of a quantum wave function, or wave functions, superpositions of wave functions in the brain, which, when we actualize a thought, for example, they collapse and become classical, triggers, mechanisms that lead to some action. So that was kind of a general, very philosophical, I would say, concept that Penrose proposed. And then I think Stuart Hameroff read the book and he said, well, I know how this could be happening. And Stuart Hameroff is a practicing anesthesiologist, and he wondered about this question for all his professional life. And he came to the conclusion that the best candidate for a quantum information processing would be neuronal microtubules. because of the regular structure. This is really the only structure which has multifunctional properties, and also the center of neuronal architecture, the center of cell division, and performing these amazing functions. But we still don't know, how it comes about, what is causing these transformations, and so on. So they started collaborating around 1993, I think, and wrote a number of papers hypothesizing that microtubules actually are biological sites for the quantum effects that might lead to cognitive processes, and etc. Now, of course, it's a hypothesis that has not been demonstrated experimentally until now, basically, when doing these experiments currently. So there's a lot of course, gaps in this. Before you reach the human brain, you have to, first of all, demonstrate that the building blocks, tubulin, protein, satisfy these conditions to at least partially have quantum degrees of freedom. Then whether the second question is, and there were many objections to it, and still are, this is in a liquid environment, so very few examples in physics can be quoted where fluids have quantum properties or objects in a fluid environment because of the noisy dynamics, basically. The second thing is physiological temperature. We live at 36.6 Celsius under normal conditions, and that's a very high temperature compared to physical experiments under controlled conditions. And thirdly, tubulin, the protein that makes up microtubules is very large compared to, let's say, quantum states in physics. So its weight is equal to 110,000 protons or hydrogen atoms. So there's every statement I made is a question mark. Why this? Why this? Can this be true? And so on. So, and that's only within the neuron. How do we then go beyond? Even if you demonstrate that the microtubules in neurons are quantum objects, how do you go beyond? But I think we have to start somewhere.
How do you link the quantum to the classical?
Exactly.
Yeah. And this is the criticism or the objection is that the body is too warm, and quantum states are, these are like a particle in a box, like Schrodinger's cat. If you jostle the box, the superposition falls apart, and that's why people who are making quantum computers are trying to cool their qubits down very close to absolute zero, so no thermal energy jostles them out of their superpositions. So you need some sort of magic to keep these things isolated in a warm, wet body to do this quantum stuff. And I think there's a lot of interesting research going on right now, and people have realized that there are quantum effects that happen in living cells. I think the original demonstration was in photosynthesis, that they've actually found there are quantum effects that are present and significant in certain cellular processes. And so this is kind of opened up a new field of research, I think.
Well, exactly. This is a perfect segue to what I was going to say. Namely, photosynthesis was studied by physicists and quantum chemists, and about 12, 15 years ago, and that led to this quantum biology paradigm being established, because it's one of several examples where you can see quantum physics at work in a biological environment, which is, as you said, it's warm, compared to physical systems under controlled conditions. It's wet and it's kind of messy, but so we don't know how to deal with messy systems. But in the case of photosynthesis in chlorophyll, for example, you have these other reaction centers which absorb photons, quanta of light, and create excited states of electrons, which are then collected and used high energy of these electrons is used to make other molecules, glucose in particular. So it's an incredibly intricate process that Mother Nature came up with to actually start all of life on Earth, because it starts with photosynthesis. The energy of sunlight coming from the sun is used to build biomass, and that is the substrate for subsequent structure formation. Primitive animals and more advanced forms and so on. So everything starts with photosynthesis, which is, as we now know, it's a quantum process. So we looked at microtubules and tubulin, and we found that in tubulin we see similar complexes as in chlorophyll, namely amino acids called tryptophans. They form a network of 8 tryptophans in a single protein, whereas in the FMO system, so it's called the FMO, in chlorophyll, there's 7 similar organic rings. And that led us to believe that this may be the culprit due to structural similarity. And then this arrangement is actually repeated many thousands of times throughout the microtubule structure. So that may be the beginning of a network, a quantum network, that communicates and shares quanta, like it's happening in chlorophyll, for example. And so now we have these experiments which are actually proving that. Although we don't have evidence yet that this quantum excitation propagates or spreads over the entire microtubule. A microtubule is the size of a cell, roughly speaking, on the order of tens of micrometers. So it's non-trivial from the quantum physics point of view. But we found, and this is now something very recent in the last year, we found that these quantum excitations of tryptophans, which are a bit like the photons captured by leaves in plants and some bacteria as well, they live actually even longer than quantum excitations in chlorophyll. In the quantum biology paradigm, the breakthrough was to find these excitations living up to about under a nanosecond, hundreds of picoseconds, which that's
very short.
yes. But it's enough to do the biological function, to perform the biological function which is required. And we found in our experiments, actually two independent experiments, one on tryptophan fluorescence, that these excitations last even longer, 5 times or more, 5 nanoseconds or more. So we are actually confident that this process is longer lasting in tubulin than in the FMO system in chlorophyll. And even longer in another experiment, which is now being performed by the team of Professor Dogario at the University of Central Florida. They illuminate microtubules with visible light and observe re-emission of this light by microtubules. And this re-emission takes place over hundreds of milliseconds to seconds, actually, which now we are bridging the gap to physiological durations, right? Over 100 milliseconds, typically the human being responds to stimuli on the order of a few hundreds of milliseconds. That's the typical response time to any sort of stimulus, visual or audio or whatever. So that would be quite interesting.
These experiments that are being done with light on microtubules, where you're shining light on it and you're measuring the time it takes for the light to be re-emitted by the tubules, and this is a proxy for the stability of certain postulated quantum states, which is kind of a key to the theory that these microtubules may be having coherent quantum superpositions that may be associated with mind or consciousness. So these experiments are doing real work trying to determine whether we don't know how these things stay stable, because theoretically they should be decohering very quickly on femtoseconds or whatever, and these things seem to be something in them is stable to timescales of picoseconds and nanoseconds, thousands of times longer than thermally random processes would otherwise maintain their energy state. So this is really interesting and suggestive. Is it definitive that this is a quantum issue from these experiments?
Not necessarily, but I think it's we don't have a working model yet. But as you said, you would expect this to be incredibly fast, femtoseconds, picoseconds. By the way, I should say that tryptophan fluorescence requires ultraviolet. It's a higher energy. It's not visible to the human eye, but it's higher energy photon. And the re-emission is faster, but still 5 nanoseconds. So it's more than required for photosynthesis, for example. Now, in the delayed luminescence, it's visible light, and it's these hundreds of milliseconds to seconds, which bridges the gap. And I want to say this is an exponential curve. So you have a bunch of photons emitted rapidly, but then some of them linger on. In other words, they create some excited state, maybe quantum, maybe not, and then they do something. They stay in an excited state, maybe interact, maybe create a collective state, as was demonstrated for chlorophyll, and then they collapse down to the ground state, and by doing so, they re-emit these the last group of photons. So we call the fast emission is called super radiance, and the slow is subradiance. And so it's probably two different phenomena, fast and slow. The fast is probably just exciting, de-exciting, staying locally perhaps. Just one group of molecules gets excited, but the slow one probably is creating a collective state, which is trapped in that structure. And that would be the best candidate for linking it to some physiological functions.
Interesting. So this is storing energy in excited electronic states. The photons come in, the electrons are excited, they store this energy, and maybe some computation happens and redistributes it in some other fashion over extremely long periods compared to what one might otherwise expect.
And one more thing, if I may add, because this is important, we also, in the Princeton experiment with tryptophans, we also probed the size of this collective state. It turns out that it's about 50 nanometers, which is about 6 dimers. Each dimer is 8 nanometers long. So that's also very significant because it's an order of magnitude greater than the FMO systems in chlorophyll. So these states seem to be longer lived and larger spread in throughout this structure.
And you've also done some work on looking at how anesthetics affect this state?
Yes. So this is the most direct link to human physiology, to the human brain, because we know anesthetics are an incredible group of molecules. They don't really have any common structural feature, but they still are able at some concentrations to turn off our consciousness reversibly. We still don't know the mechanism. There are some, hypotheses, and all of the hypotheses so far have some holes in them, and they have to do with the membrane's receptors in neurons. But there are many holes, and Stuart Hameroff, being an anesthesiologist, he wrote a lot about these different hypotheses missing the mark. And in the case of tubulin, we know that anesthetics binds to tubulin, and this was independently confirmed by the work of Professor Eckenhoff at the University of Pennsylvania. And tubulin and actin, I think, are the only two proteins which bind all anesthetic molecules known to us. And so there was one interesting connection, but only correlation, not causation by any means. And we worked in these experiments that I mentioned to you at Princeton and University of Central Florida with two different anesthetic molecules, trying to see if they affect these processes that I told you about, the quantum absorption, emission, photon. I would say photon, but photons are quanta of light, so I guess I'm allowed to say quantum. And and the results are not overwhelming, but statistically significant. So that means that in both cases, we use isoflurane and atomidate. In both cases, the lifetimes of these effects are shorter. In other words, anesthetics make this process, they abrogate to some degree. We haven't done dose response experiments where you change the concentrations of anesthetics. We try to use something close to physiological. So the reduction in the lifetime is about between 10 and 20%. So that's basically where we are at today.
So that's interesting. So this is an experiment that it doesn't prove anything per se, but it adds evidence. It was a chance to falsify one of the key assumptions behind the orchestrated OR, which it needs to show some sort of response, or the theory just doesn't work. So this is positive for the theory, I guess. What's next?
Exactly. And I should also say that these experiments and this entire project has been funded by the Templeton World Charity Foundation. So we acknowledge that support. And the stated objective was to falsify OR, to demonstrate that there is no quantum. First of all, there is no quantum effect. And secondly, that there is no effect of anesthetics. And we were not able to falsify. It does not necessarily support 100% the predictions of OR. And I think we ended up believing that there is strong support for quantum effects, which are potentially long enough and potentially large enough in size, and also that they are affected by anesthetics in the correct direction. We don't know if this explains anything, but we say that this provides feasibility or plausibility of the assumptions. And in order to go next, that's to answer your question, we need to do this in a living environment, because that was under controlled laboratory conditions. We need to now test something like this for a single neuron that is alive. So that's the logical next step. And we're now writing up peer-reviewed papers and reports based on what we've done so far.
Wow. So that's an ambitious next step.
Now we will be getting to the questions of quantifying it. Namely, the answers that I provided so far to you are, I would say, largely qualitative. Long times, large range, reduction of the effects by anesthetics. But now the question will be, if you have a neuron, is the amount of photon absorption emission compatible with what we know about living cells? Do they actually, we don't have lasers in our brains, of course. So, but now the question will be, where are these photons coming, if they are, or if they are coming, do we have a sufficient number of photons to create these quantum states and maintain them, and so on and so forth. So the next group of experiments, whether we do it or somebody else, will have to be bringing this close to biological reality.
So does this theory suggest that photons are somehow being captured in biology and funneled through these cells in some coherent state? So these are ultraviolet photons, you said, invisible photons that are participating in this process. Are they key to this?
Both experiments deal with different frequencies. Tryptophan fluorescence deals with ultraviolet photons. The delayed luminescence, where you have longer-lived states, deals with visible light. The question about creating these kinds of photons in the human body is an open question, because we know only from observations of emission of photons from the surface of the human body, the brain, a very low intensity that has been measured and might not be enough. But we don't know if photons are emitted and reabsorb inside the body, and they never. What percentage of these photons are escaping to the environment? We don't know that. So it's quite possible that there is a sufficient number of photons generated internally and used for this kind of cognitive activity, and that would actually go along with Albert Buehler's work, who postulated actually two things. The first one was that mitochondria, by producing energy, biological energy, or transducing, I should say, biological energy, providing ATP molecules to drive all the metabolic processes, as a byproduct emit photons largely in the sort of range that could be utilized for this. And secondly, the question is, what is receiving them? So here could be microtubules, but Albert Buehler stated that in general cells could have a perception mechanism for incoming photons in the form of centrioles. So that opens another direction, but I don't want to kind of muddy with the waters. Just to say that the role of photons may also be multifactorial, one of which would be to use as fast mechanism of some computation, let's call it this way. Another might be for recognizing living systems in their vicinity, for example, other cells. This is exactly what Albert Buehler did. And there may be some other ones driving other biochemical reactions. So biophotons is a subject which has a long history, but very little solid science behind it. So I don't want to engage in too many hypotheses.
The more I learn about this, the more I realize we'd have just scratched the surface, the barest surface of the biology of just the individual cell. It's really amazing. I know I had an earlier interview with a philosopher, Arthur Reber, who believed that cells were conscious, basically that consciousness started in cells and evolved, billions of years ago over, the process of evolution is kind of a trial and error. How do living cells best adapt to their environment? So they learn things through trial and error over billions of generations. And if there was something here, then it's very possible that, biology has learned how to use it, and that's become a basic element in all cells. And so if we posit that individual cells are conscious, I mean, taking a step back from all the confusing physics and biology, how do you move beyond individual cells to groups of individual cells creating a conscious mind?
Yes. I wish I knew the answer.
And where do you stop?
No, but I think it's quite plausible that the connection has to do with synchronization, some sort of synchronization of activity, synchronization of purpose, collective behavior, and I think that's what our human bodies are very good at, because a lot of synchronization is taking place. And actually, Dr. Dogario from the University of Central Florida has performed such experiments, others as well, where they demonstrated that cells synchronize their activities in cultures, for example, without chemical signaling. Dr. Dogario performed experiments where groups of cells were of the same type. They were held in one part of the Petri dish, separated by some kind of plastic wall, I believe, from another part, and one part was given nutrients and upregulated their activities, and the other didn't, and they also upregulate activities as if they were being sent signals from those that received nutrients to increase their metabolism. And this is one example. There were similar examples by Daniel Fels from Basel in Switzerland, who showed, I believe, cell division was synchronized by physical means, not, let's say, chemicals being released and captured by the other ones, which also happens, I think. But there are experiments which show And so what is physical that can do this? And I would say electromagnetic. That's the most obvious answer to it, synchronization. And that probably also brings, again, Mike Levin's work, who believes in electrical, synchronization and programming of cells. So I think we have a long road ahead of us, but we have to open our minds and allow these new questions to be asked and gradually answered. We worked with several frequencies of electromagnetic and electric fields, and each of these frequencies provided different insights, different effects were observed. We worked with terahertz, for example, and just published recently this year a paper showing that with terahertz radiation, which is much lower frequency, basically microwaves, you could break microtubules into pieces over about 15 minutes. So where is this energy going? What's happening to this electromagnetic energy, which is below the thermal energy, KT, which is everywhere for free, more or less. So that's one example. With tryptophan, we looked at ultraviolet and nanosecond states with visible hundreds of milliseconds. So the question is now, how do we study this with very systematic approach? And I think that also indicates there are, if not resonances, that there are probably bands of frequencies which do different things, almost like different communication channels. Dial on the radio, you can change from AM to FM and go from kilohertz to megahertz, and you listen to different stations. So
I want to poke at one thing you just said. Our microwave frequencies can break apart microtubules. This is this related to the ongoing controversy over ill effects of RF fields? There's a large community of people who believe they're affected by RF fields in health ways.
Well, yes, possibly, but so the devil is in the detail, and the detail here is twofold. Number one is the duration of exposure. So we needed at least 15 minutes to see disassembly of microtubules. So that's a long time of exposure. And secondly, high intensity. And again, if we drop the intensity of this terahertz radiation below a certain threshold, nothing happened. So you see that critical parameters, and I think you can introduce a lot of fear into people's minds if you don't pay attention to the critical parameters. If it's low intensity, nothing will happen. And the third thing is penetration depth. So this may not go under the skin, so to speak. It has to be something which is exposed to it. And of course, the deeper it goes, the lower the intensity. Therefore, even if the intensity of that particular radiation is high enough to do damage, but as it goes through the skin, it'll diminish and nothing might happen. And by the way, terahertz radiation is used in airport scanners, security scanners. So there was a lot of backlash against it, but so far I don't see any evidence for some adverse effects.
So you carry your cell phone in your pocket with no problems?
Yeah. Maybe too much.
Very interesting. So I think we're kind of reaching the end of our time limit here, but it's been really enlightening to speak with you and learn about all of these interesting physical processes that are happening in the subcellular region that could be linked to consciousness. I love, a lot of people don't like not knowing and not having certainty, but I love the open fieldness of this investigation, and there's so much that we have yet to learn. This is my favorite part of science is when you can start putting out all of these interesting ideas and testing them and doing real work this way. So really great to have you and to learn all about. I have to look now into optical biophysics and
I couldn't agree more with your comment about being attracted to things which you don't know. That's why I think I'm a scientist, and I guess you're a scientist at heart. If you're not doing science, I'm not sure, but you should be.
Yeah, thank you. So before I let you go, I'm going to send you a Rational View t-shirt for coming on the interview. Really appreciate you chatting with me, and thank you. If you'd like to follow up with more in-depth discussions, please come find us on Facebook at The Rational View and join our discussion group. If you like what you're hearing, please consider visiting my Patreon page at patron.podbean.com/TheRationalView. Thanks for listening.